Alrighty, here comes video number 2. Um, the second part of protein synthesis is translation. The first video was dealing with transcription on how the information from DNA that is in the nucleus can get copied into a RNA molecule, and then how that RNA molecule then is going to leave the nucleus by being processed, and it will go into the cytoplasm and it will find a ribosome. And that's when translation will begin. Remember, transcription occurs in the nucleus because that is where the information is on DNA. DNA never ever leaves the nucleus, but proteins are made outside of the nucleus at the ribosome in the cytoplasm. So how do you get the information from DNA in the nucleus to the ribosome outside of the nucleus? You do that through an RNA molecule. So RNA is kind of like our middleman. And so RNA is transcribed and produced during the process of transcription in the nucleus. Once it's ready to leave the nucleus, it leaves through a nuclear pore, and then translation can begin. And so you're going to see some things here that are going to be similar to transcription. This is another video that I would like for you to watch on your own time. I'm not going to take the time right now to show it to you, but you can watch it. The videos that I've uploaded serve to pull it all together, okay, or to give you an overview of it. All right, so before we get too deep into this process, translation is going to have some players involved. And the first one obviously is going to be mRNA that is carrying the message. That's why it's called mRNA. And then you've got tRNA, which is going to be the translator or the interpreter of the message. Remember, I said in the last video that we're going from one language to another language. We're going from nucleic acid language into protein language. And so tRNA is going to be the interpreter. And then you've got the ribosome, which is where it's all going to take place. And you've got amino acids that are going to be brought to the ribosome. And that's where tRNA is going to come into effect there too. tRNA is going to be like the taxi cab that is going to pick up the correct amino acid and bring it to the ribosome in the correct order. This is a summary of translation, and this is probably going to make a little bit more sense once I'm finished. But here is a ribosome. You can see that it's got 2 parts, and there are 3 sites, the APE sites, or you can go left to right, EPA site. The reason I go from right to left is because the A site is, and the P site is where we're going to start. But anyway, here is your ribbon-like RNA molecule, your 5' cap and your 3' or your poly A tail here. And the 5' cap, remember, identifies the ribosome is going to be able to recognize that 5' cap is where it needs to attach to begin with. And then you can see tRNA is bringing in these amino acids right here, and they're bonding with the 3 base codes that are on RNA. All right, like I said, that's probably something that will make a little bit more sense at the end. All right, so remember the players. mRNA is transcribed in synthesized, in transcription. It is composed of codons. Codons are 3 base sequences of mRNA that code for amino acids. So this is an mRNA molecule that is processed and ready to go into the cytoplasm. And you can see right here that you need to read this in 3 bases, in segments of 3. So like CAG, AUG, GUA, CGG. those 3 base segments are called codons, and that is going to be very important whenever you're learning to translate a protein. You're going to need to use these codons in the chart for the genetic code. There are specific start codon, or there is one specific start codon called AUG, and that is the very first codon for the gene on the mRNA, the coding sequence. And then you've got 3 different stop codons, and UGA is one of them. Okay, another player is going to be the ribosome, which is made in the nucleolus. It's made of rRNA, and it's got 2 subunits, large and small. Kind of looks like a hamburger bun, if you use your imagination. And then in the large subunit, you've got active sites, and these active sites are the A site, the P site, and the E site. And E stands for exit, and so that kind of lets you know that that's going to be the last site that tRNA goes in. The A site is the one where the amino acid is going to be added to. And there's that picture again. tRNA is transcribed in the nucleus. Again, that's not really that important. It is specific to each amino acid, and what it's going to do is transfer the amino acids to the ribosomes. They're the taxicaps. And what is unique about tRNA is it also has 3 base codes called anticodons. On one end of the tRNA molecule, this funky looking shape here is tRNA. On one end, it's got the 3 base code that is called the anticodon, and it is going to pair up with the codon. And so on the other end, up here, on the 3' end, you've got the place where the tRNA is going to pick up an amino acid. So AAG is going to code for, it's going to pair with a particular codon, UUC, on the mRNA molecule, and that is going to tell the tRNA which amino acid it needs to pick up and bring to the ribosome. The base pairing rules between 3rd base of codon and anticodon are not as strict. This is called a wobble, and I'll get to that a little bit later. We'll see that in just a few minutes. The enzyme that is going to bind tRNA to the specific amino acid is aminoacyl tRNA synthetase. All right. There's 20 amino acids. They're held together by peptide bonds. This is all review information. And like I said, you have to translate, go from one language to another. Nucleic acids have a 4-letter language, A, T, C, G, or AUCG, while proteins have a 20-letter language, meaning the 20 different amino acids. So if I put if I make one base code for one amino acid, that only gives me 4. If I have 2 bases coding for one amino acid, that only gives me 16. But if I have 3, that gives me 64 combinations that can code for 64 different amino acids. Well, we only have 20 amino acids. So then that means that we're going to have some amino acids that have more than one code. Like phenylalanine has these 2 right here. Serine has these 4 and these 2. So it's got 6. So anyway, that is more than enough coding capacity for what we need. This chart is what you will be using whenever you're building your own proteins, whenever we're doing practice. Okay, so steps, initiation. The small subunit is going to bind to the 5' cap at the start codon where AUG is. So AUG is right here, and the tRNA is going to come in. The tRNA that has UAC as an anticodon will come in, and it will bring the amino acid that codes for AUG. Now, AUG is on mRNA. Remember, the codons code for amino acids. So if I go back up here into my chart, I can find AUG, and that is the amino acid methionine. tRNA carries the amino acid, and it attaches to the P site. That is where the AUG will be. It will start in the P site, but look, it leaves the A site open, and so the next amino acid will be brought, or the next tRNA will be brought into the A site, bringing that next amino acid. The large subunit attaches, and now we've got like a literal hamburger. The mRNA would be like the hamburger meat in between the two parts of the ribosome, the bun. Here's a bigger picture of what I just showed you. Okay, so then elongation begins, and you've got the second tRNA is going to come in, bringing the second amino acid. And like I said, this is codon recognition right here. Here is a elongation animation that's going to show you specifically how this works. I suggest you looking at it. It's just a few seconds long. But peptide bonds will be formed. Here's the second amino acid coming into the A site. This is tRNA. There's the amino acid coming in. And then a bond is going to be formed in between these two. That's the peptide bond. And you can see that we've got a growing polypeptide right here. And then elongation continues with translocation. Translocation just means that the ribosome is going to move down to the next codon, and that means that whatever is in the P site will now move to the E site. Here we are. The P site will move into this tRNA will move into the E site, and it will exit. It will dislodge or unbond from its amino acid because the amino acid is bonded here. And then what was in the A site now goes into the P site because this whole ribosome is moving down. Again, I recommend that you watch this animation because it shows it very well. It's just a few seconds long. And then this is going to continue to happen until the ribosome reaches a stop codon. And here are your 3 stop codons that you just kind of need to memorize. And translation stops here. A release factor, which is another protein, binds to the stop codon in the A site. There is not an amino acid for a stop codon. There is an amino acid for the start codon. It's met. But for stop codons, you just have this release factor that binds with it in the A site, and then that lets the ribosome know, okay, we've done our job. We have created this protein or this polypeptide right here, and now we're just going to break apart. We're done with our job. This polypeptide is not a protein. It's got to fold and twist and become an alpha helix and a beta pleated sheet and tertiary structure until it gets into the shape of the protein that it's supposed to be. Here are some animations for you as far as termination and another animation that will go with translation. And I hope those links are set. If they're not, I will give you some other animations. Polyribosomes just let you know that many ribosomes can translate a single mRNA. So the advantage of that is that you can get lots of proteins being produced at one time from one strand of mRNA. It's pretty advantageous. Prokaryotes versus eukaryotes. Just look over this. It's very simple and cut and dried. And then this is where I'm going to stop this video because the next video will be on mutations. Hope this was helpful.